Yeah, it's a pleasure to be here. I'm Morgan Hocking, a senior environmental scientist with Ecofish and adjunct professor at University of Victoria. And I'm excited to be talking to you today about a fun collaboration that we've had with the Helbing and Lesperance labs and grad students at University of Victoria, as well as with the commercial eDNA lab Bureau Veritas in environmental DNA methods development and application in environmental consulting. So as you might tell from the name Ecofish, we do a lot of fish population assessment and monitoring using traditional methods. We work primarily in British Columbia, but we're growing rapidly across Canada, and we service clients in the hydroelectric industry, in mining, with government, First Nations, and various other industries and communities. As has been said many times in the introductions to many eDNA papers, there are a lot of challenges with traditional fish monitoring, and including missed detections and challenging environments to sample in. And there's certainly many opportunities to add eDNA to our toolbox in supporting environmental assessment of fishes. And so to do that, Ecofish has been partnering with UVic for a number of years. We've received a number of grants. MyTax Accelerate, a collaborative research and development grant. You guys heard a bit about the Karut project, and I'll talk a bit about that project in a sec. We're also a collaborator on the iTrack DNA program, and we just received a Karut Part 2, actually, through the BC Salmon Restoration Innovation Fund to monitor oolakan outmigration in the early marine environment, which, including with eDNA, which is a very exciting project. So UVic has been developing the targeted assay tools for monitoring priority fish species, and we've been validating their use at Ecofish monitoring sites, as well as I'll talk a bit about this paper we published where we did this experimental validation in hatchery tanks at the Goldstream hatchery. Also point out that we used the Hocking backpack there. For our sampling. We did start by using bottle samples, but we've been finding this very effective sampling program. I'll also point out that Bureau Veritas and UVic have been collaborating for a number of years. This started in 2017 through an Innovate BC Ignite grant, and they've been doing various collaborative activities to validate assays and compare assays between labs. Ultimately, Bureau Veritas is validating and commercializing the eDNA tests that are developed at UVic. And this is very helpful for us. When we started our eDNA programs, we were only sending our samples to UVic. And now, since it's expanded rapidly, we're sending practically 95% of our samples to BVL. So through this collaboration, we've been developing and applying Canadian standards for use of eDNA in environmental assessment. We've contributed to the CSA standards that Ken was talking about yesterday. And some of the key questions we've been working on include species detection at low density, estimation of species abundance at both high and low copy number. Some of the aspects that Matthew Yates was talking about yesterday, the influence of habitat, such as flow, on species detection. I'm going to talk a bit about some work on incorporation of environmental or false positive background into species detection. And then one of the new things that I think we'll be working on a lot, especially as applied to mining industry, is this influence of inhibition on estimates of copy number. We're finding this to be incredibly important. So just to talk a bit about a few projects that we're involved in. You heard some from those speed talks on Sunday night about the Karut program, a very exciting multidisciplinary program in collaboration, the Conservation and Recovery Research on oolakan and Haisla Territory project to assess oolakan population status, health, and stressors and options for recovery in support of the Haisla Nation. Students Michael Allison and Meredith Pochard are involved in this program. Thank you very much, Michael, for developing an incredible assay for oolican. It's working extremely well. Just show some of this data again from this program. This is some sampling that occurred in 2019 at the Camano River, monitoring both eDNA signal as well as egg drift after spawning. And you can see, so this is daily sampling across roughly a 2-week time window. You can see 0 signal at the start of the monitoring period, and then you see a hit with eDNA before the observations of oolican in the river and the egg drift. And then we see this incredible spike of eDNA signal actually timed to the spawning event, and then also a decrease in the signal through time as correlated to the egg drift. So I'll show you this spike as well. This is the same data, and above and bottom panel is the same data as well, just a different y-axis scale. And we can see this spike of the spawning event that occurred on the 24th, up to 75 million copies per liter estimated at flowing down that river. And we can calibrate to flow, we're doing that as well. But this is a way to potentially estimate stock biomass as well as to look at run timing in the river and particular spawning timing. So yeah, this work is going very well. We've expanded our programs to a number of different watersheds along the coast. We have, for example, a long-term program started on the Skeena River. This is also a very exciting project, interesting project working on the Fraser River. This map shows, this is essentially Vancouver here in Richmond, the mouth of the Fraser River, some of our eDNA monitoring sites on the Fraser for oolican. Essentially, this project is about determining run timing and relative abundance of oolican from eDNA to inform work windows for a potential large infrastructure project. The twinning of the George Massey crossing, essentially a tunnel underneath the Fraser River at George Massey, just south of Richmond there. And so the idea is to use eDNA to inform this project and the work windows for construction for this project. So just some preliminary data here, just collected this year from early November through to just a few weeks ago in mid-May 2023. We're seeing some really interesting results from our monitoring at George Massey and just above at Petula Bridge. We're seeing the first detection of oolican into the Fraser River in late November, which is much earlier than has been detected previously. We're also seeing this bimodal distribution of oolican signal through time that is suggestive of potential early run, followed by the main late run that's well known in late April and early May. And so this is really interesting stuff, and it's also this possibility of 2 runs aligns with indigenous knowledge in the area. So that's just a little tidbit of some of the oolican programs we have going. I wanted to talk for the rest of my talk here about some of the species detection methods, particularly at low copy number. I think some folks have heard about the EloQuant quantification method for estimating species abundance at low copy number. Wanted to emphasize the importance of Mary Lesperance, the lead author of this. She's a statistician at UVic who's kind of led the mathematical theory and R-code development for this. It's been a pleasure to be involved. I think folks are fairly familiar with these standard curves, in particular at high copy number between CT and copy number. What's novel about this is the low copy number regression curves using a binomial Poisson model or standard curve for low copy number, which also provides the assay limits of detection, which account for the false negative rate. And a great feature is that these LODs also decrease as you increase your sampling effort or the number of technical replicates run on your samples. I'll just point out also that this assumes a limit of blank of 0. So essentially no contamination. And so we have now produced a follow-up to that paper, the species detection at low copy number with an environmental background, and so to account for false positive error. And this was developed through this experimental test with two different experiments. One at high copy number is this density experiment where we added fish to these 10 juvenile co-salmon actually, to these 10,000 liter tanks, and looked at the relationship between COA biomass and eDNA signal, as well as the dilution experiment, which was our low copy number experiment, where we had 3 fish in a tank, and then we removed them and ran flow through water and looked at the dilution of the signal through time. And one of the interesting things, I don't know if you saw that flash up there, but we actually had, kind of by accident, an environmental background that was part of the study, and we're not totally sure how that gets in there. We did clean all these tanks extensively, but with what we learned earlier today about eDNA in the air, it's this is a hatchery, so we are seeing an environmental background, and so this dilution experiment is really nice to see how it dilutes to the background and at what level of dilution is it distinguishable from the background. This is our result at high copy number. I won't go into this a ton, but this has been shown a fair bit in the literature, but we're really obviously happy to see this type of relationship. In terms of the, just a bit of background about false positive and your environmental background, we do see this a fair bit in our studies so far. And so, false positives can be caused by contamination in the field in the lab, assay non-target amplification, also like natural contamination or spatiotemporal dynamics of eDNA. So, for example, we could be monitoring a fish reach like above a barrier, but maybe bears or birds can distribute the DNA upstream. So a low detection rate, when we're trying to detect DNA at very low species density, that needs to be distinguishable from this background false positive signal. So, of course, field blanks and field negative control sites are crucial for that interpretation. And so this is, the results of our dilution experiment. And essentially what these figures are showing are the results of the estimated copies, estimated from eLequant, across that dilution series. And then this is, you can see the background signal actually is indicated by that fish in the solid dashed line. And so you can see the signal approaches the background after 80 kiloliters of dilution. But the point of this is more about how it illustrates the method. And so the eLequant script was adapted to account for the environmental background signal. And what you get is an updated limit of detection and limit of blank with background, which is ultimately a test that the signal that you observed is different from this environmental background. And so the test result is positive if the LOB, if the result is greater than the LOB-B, but uncertainty remains if the result is less than the LOD-B. And so we get these different ranges. So we're saying a detect above the LOD-B. If it's less than the LOB-B, then it's a no detect. And if it's in between the two thresholds, then we're calling it a detect star. Which for us, we're kind of describing this as target DNA is detected with uncertainty, and more sampling is recommended to confirm the result. And so showing this 8 versus 24 technical replicates shows how is if you do more sampling, it lowers the LODB and LOBB, and you get more confidence in the result that you observed. So I think I need to wrap up. We did have an example I could show of how we're using this in like fish presence assessment in streams, mine sites. Ultimately, the main message is that we need to measure the environmental background, and eFish is one of the assays where we're consistently seeing an environmental background. And so then using this background from our negative control sites, we update our limit of blank and limit of detection with background, and then can interpret our test sites, relative to that LOB and LODB. So I'd be keen to get some feedback on this method and see if folks would be keen to adopt this approach and account for environmental background in the same way that we have been doing. But we've been needing to do this because we need to give clear scientific advice to regulators and have like a clear, repeatable method to account for this false positive background. So this is just a similar example. We like to run many assays on these sites, and this is just an example. It's much simpler where we have no background to account for. So yeah, just key takeaways. It's been a pleasure collaborating with UVic and BVL and developing eDNA methods through that work. We now have like 15 plus active projects related to eDNA monitoring using assays developed at UVic and commercialized at BVL. And I think these methods, standardization methods, are crucial for continued adoption in environmental assessment. So yeah, just wanted to put a plug out there for if you wanted to join Ecofish, and we're growing and our eDNA programs are expanding. And yeah, thank you very much.